Okay, good morning, everyone. My name is Yuichiro Tsuchiya from the Institute of Transformative Biomolecule in Nagoya University in Japan. And thank you for interaction, and thank you for inviting me to present our work related to parasitic plants. So first of all, I'd like to introduce our institute. What's Institute of Transformative Biomolecules? So the concept of our research institute is the collaboration between chemistry and biology. So we are trying to work on that area in between chemistry and biology. So one of the part is to develop fluorescent molecule that makes cell visible. And of course, another area is to develop a small molecule that regulates plant growth or animal growth. And also, our chemist is trying to evolve their method to efficiently synthesize small molecules. Okay, so here is how our institute looks. So the upper part is an office area, and the below part is a laboratory area. So, okay. So this is how it looks in our laboratory. So here is a draft. So chemist synthesize the molecule here and bring the molecule to the table, and now biologists test on a like cell-based assay and give feedback to the chemist and synthesize the draft just behind. So that makes the collaboration smooth and fast. So I'm really happy about that environment. And one of the area we are interested in is related to parasitic plants. So parasitic plant, it's a higher plant, but it somehow evolves the way to take nutrient from other plants. So in this case, the one we are mostly focused on is called Striga helmontica. So this shows a beautiful pink flower above ground, but below ground it attached to the host plant like crops. So here is how it looks. And the seeds are tiny, so it can spread easily. And now the parasitic plant is causing huge problems in African countries. So here is a field in Sudan. So this is supposed to be a field of sorghum, but you can see all the pink flowers blooming. And afterwards, the striga killed the sorghum. So eventually so the farmers can't harvest any grains from this field. So the striga, it's believed that striga is originated in a north east part of Africa, but it's now spread to the most of the African countries and causing $1 billion of damage for every year. And it's believed that 100 million people's lives are affected. And because the damage is so large, and the United Nations now declared the striga problem as the largest impediment to poverty alleviation. So not only in African countries, but South Asian countries, including India, have the related parasitic plants called Orobanche caused problems on crop productions. For example, here is Orobanche aegyptiaca. This have wide host range for broad leaf crops like tobacco and mustard and tomato. And those two related species also exist in India and causing damages. So it seems like 50% of the area in a particular region is contaminated with the seeds is contaminated, and that leads a yield loss for the 50% to 60%. So it's not only the problem in Africa. So our purpose is to develop a small molecule that makes the pink field back to green. A key step for the parasitization is germination. So it's mediated by a plant hormone called strigolactones. So strigolactones are the molecule as indicated here, but it doesn't indicate a single molecule, but related the molecule with the related folding structures. And so there is some differences, like OH group indicated in the first ring, or the another strigolactone have an OH group in different rings, and they are all called strigolactones. So in host plants, strigolactone function to suppress shoot branching and root response, and also for abiotic stress seeds. But unlike other plant hormones, the strigolactone is released from the root to the soil and function as an ecological signal. So the released strigolactone is perceived by a symbiotic vascular mycorrhizal fungi, and the fungi grows toward the host root by sensing the gradient of strigolactones. Now, Striga seeds, so the seeds are unusually, unlike other plants, the seeds are just super dormant, and they never germinate in a soil. However, when the host plant grows around, then the Striga seeds detect strigolactone and start germination and proceed for in parasitic processes. So the strigolactone had discovered at 1960s, but since that the mechanism of strigolactone have been a central part for the Striga research, because the germination stimulant like strigolactone can be used as a suicide germination. So as mentioned, Striga seeds never germinate without strigolactones, but once the seeds are force-free germinated by strigolactones, then the Striga seeds just died because they obligate parasite and they can't survive without host plants. So, to make the suicide germination, we need to develop good germination stimulants, because we can't directly use strigolactone as an herbicide. So it's too expensive and it's unstable. So before that, the chemists, the organic chemists have tried to make mimics for strigolactones. But before that, we have no information about how Striga respond to strigolactones and germination. And that's, we thought that's an important point for development of the herbicide. So we want to develop germination stimulants. So to develop that herbicide, the most important information is the structure of strigolactone receptors. But before that, strigolactone receptor itself was unknown in Striga. So we needed to identify strigolactone receptor first. So to identify that, we have, in collaboration with chemists, we developed fluorogenic strigolactone mimics that can probe the function of strigolactones. And using that, we successfully identified the strigolactone receptors in Striga, and also crystallize and solve the crystal structure of strigolactone receptor in Striga. And now we are using that information to develop a suicide germination molecule. That's the overview of our research. Okay. So just briefly, I wouldn't give too much details, but since strigolactones were identified as a plant hormone, so now we can use genetics to identify components related to the strigolactone signaling or biosynthetic process. So those efforts have identified components for strigolactone signaling. So here you can see on D14, this is putative, it's an strigolactone receptor. So that mediate that replace shoot branching through the ubiquitin-dependent proteolysis of the repressor proteins. So, but seems like the strigolactone in non-parasitic plants or host doesn't have roles in germination. But what's interesting about that pathway is the germination is mediated by a parallel pathway. So that also shares a component of ubiquitin ligases, but the receptor is different. So it's the receptor is called Chi2 or HTL. That's a receptor for not for strigolactone but for germination stimulants discovered from smoke-derived germination stimulants called caricin. So that's in non-parasitic plants. So we initially looked for receptor using database for Striga cDNA, and I found several genes have a related sequence to D14 receptor, that's for branching, or HTL, that's for germination in response to caricin. So here is a phylogenetic tree for that. So as you can see, so the gene indicated by red is a gene for Striga. So as you can see, there is only one homologs of D14 for shoot branching, but you can see like the receptor related to germination is amplified at least 11. So now we thought that could be a good candidate for strigolactone receptor. But the problem is for studying Striga is that it's impossible to do mutational studies because we can't. It's really hard to grow the parasitic plants in a laboratory. So need to parasitize the host, and also this the Striga is outcrossing species, so it can't generate seeds by itself. So it means that most of the genetics analysis is impossible in Striga. So we can't knock out the candidate receptor. Also, Striga is not, we can't transform Striga, so that's also a big issue. So as for biochemical study, so we have now 11 candidates and several strigolactones, and testing the binding for the combination of those is really hard. So that's the problem we had, although we found a good candidate. So our chemists have solved this issue by developing a fluorescent molecule. So, I forgot to mention that strigolactone receptors are related to alpha-beta hydrolase. It's related to enzymes, but also function in signal transductions. So unlike other receptors for plant hormones, so this receptor for strigolactone have unique characteristics because so strigolactone receptor perceives strigolactones, but afterward it hydrolyzes strigolactone into 2 pieces. Okay, so our chemist took this reaction to design this kind of molecule. So this also functions as a strigolactone, so it's perceived by strigolactone receptors. But after perceived, it's hydrolyzed into 2 pieces, and one of the pieces becomes fluorescent. So it's a kind of fluorescent turn-on molecule. So so we took this molecule to characterize our candidate receptor for strigolactones in Striga. So what we first do was a competition assay. So now just summary. So here is the candidate for the receptor, so it's hydrolyzed strigolactone and become fluorescent. But when in a test tube, when the competing molecule that binds to the pocket exists, then the fluorescence shouldn't occur. So using that competition assay, we were able to prove that the candidate actually binds to strigolactone. So in this case, we used strigolactone receptor from Arabidopsis. But you can see the dose-dependent decreasement of the fluorescence by adding synthetic strigolactone called GR24. So by that, we can now measure the IC50 value for the relative strength of the binding. So however, this the molecule called Carba GR24, that's the difference for those two. It's only this part that have oxygen active, but this Carba GR24 have carbon in this part. That small difference makes this molecule inactive. But using that molecule, we are not able to see the binding. So our competition assay competitive binding assay system seems working to detect binder and non-binder. So we tested using that competitive binding. We now looked at the binding among several strigolactones against 10 of the 11 putative strigolactone receptors in Striga. And here is how it looks. And so it's kind of busy data with the graph. So I summarize as a heat map. As you can see here, it's oops, just a little bit off slide. So here is receptor for each receptor, and here it's the strigolactone species, and red indicates high affinity, and white indicates no binding. So as you can see that each strigolactone receptor have different preferences for strigolactones. So for those two, that binds to all the strigolactones, but this one doesn't bind to orobanche, but this one binds to orobanche but not strigols. So although those receptors have different preferences, so those 8 have binds to different strigolactone with high affinity. So probably, so although there are many, but probably those 3 receptors are the most important one for target. So we also expressed individual strigolactone receptors into Arabidopsis and see whether Arabidopsis become responsive to strigolactone in germination or not. So in that case, this axis indicates the each Striga strigolactone receptors expressed in Arabidopsis and tested for those molecules for germination. And as you can see, the group of these receptors gives Arabidopsis to respond to many different strigolactones. And among that, the one we call ShHTL7 gives Arabidopsis to sense strigolactone at picomolar range. So it's a very, very high sensitivity expressing ShHTL7, but TN1, that particular strain doesn't really stimulate germination. So that's quite an interesting way to use that Arabidopsis strain as a biosensor for strigolactone. And also that fluorescent turn-on system enables us to see whether strigolactone is perceived within a Striga seed. So we put the Striga we give the Striga seeds with the fluorogenic probes and see the time-lapse movie. So here is how it looks. It's a bit dark, but So the fluorescence indicates where strigolactone is perceived. Let's see one more time. So here is Striga seeds. So you can see the fluorogenic probe is perceived in a root tip and makes like a wave and disappears. And again, so the perception occurs and start to the root in elongation occurs. So it seems like the perception of strigolactone Striga is quite a dynamic process. So we don't quite understand what does it mean, how relevant physiologically, but I think it's a good reporter to characterize a molecule like germination stimulants or inhibitors. So now we have set up a good system to characterize strigolactone receptors. So we now trying to identify the good germination stimulants. So we took chemical screening. So we simply screen the chemical germination stimulants for Striga from the chemical library. And from 12,000 synthetic molecule library, we have identified about 20. So that's all have related structure like that. So now our chemists have synthesized over 100 analogs. And here you can see two different assays. Below is a binding, in vitro binding to the 10 receptors. And above is Striga germination. And again, the red indicates binding. So as you can see, many of our analogs actually bind to the HTL7. So this is HTL7. Just the text is slightly moved. So many of our analogs selectively binds to the HTL7, and those that binds to HTL7 tend to stimulate germination. So we think those analogs are selective agonists for HTL7. And we have made several rounds of improvement, and actually it was quite successful. And the best candidate now shows germination at femtomolar range to Striga germination. So that's to compare to GR24 synthetic strigolactone, it have about 10,000 times of activity. So this is quite a highly active one. And again, we also tested for binding with strigolactone receptors, and it still retains a strong selectivity to HTL7, which is good. And most importantly, the molecule doesn't bind strigolactone receptor in Arabidopsis. So as discussed yesterday, this could be a good estimate for a biological safety issue. So we are also testing the effects on avascular mycorrhizal fungi. So hopefully, our molecule doesn't mess up the physiology of that fungi. So we now have a good candidate and trying to move our research to the field level, but we have just started. But before moving to the field level, we set up a system to see suicide germination effect in a pot. So this is quite a simple experiment. The Striga seeds are mixed in a soil and corn were grown. So when treated with DMSO as a control, so now you can see many Striga appearing from the root of the corn. But when treated with our molecule, the appearance of Striga is strongly suppressed. So it seems like we don't quite know what's happening in the soil, but it seems like suicide germination is really working well. Okay, that's about the story. And here is a team Striga. So we have members of the chemist from Professor Oi and Professor Itami Kenichiro. and Dr. Kinoshita Toshinori from the biologist. And we are also collaborating with University of Toronto Peter McCourt's laboratory members. Thank you very much for attention.